So good afternoon. Welcome to everyone to our departmental seminar. We are extremely grateful that today we are having Dr. Rajesh Dutta as a speaker in our departmental seminar. So let me briefly introduce Dr. Rajesh Dutta. Dr. Rajesh Dutta joined IISc in 2012 as an integrated PhD student in chemical science. Then he joined as a PhD student in 2014 under the supervision of Professor Weizmann. And 2019 he submitted his PhD thesis. So currently he is a postdoctoral research associate at the Weizmann Institute of Science now, and he will be moving to Purdue University. So he has published a total 9 papers in peer-reviewed international journal. So with this brief introduction, I invite Dr. Rajesh Dutta to deliver his
Thank
lecture.
you for your kind introduction. And so today I'll talk about understanding transition path dynamics for protein folding and unfolding. So as the title suggests, there will be a brief discussion, and I'll describe how this transition path and how one can define this transition path. And protein folding and unfolding is considered as a barrier crossing process, where like two minimas are there. One is folded minima, another is unfolded minima, and this passes through molecule passes through a barrier. And I will mainly discuss about the dynamics around this barrier, and I will try to explain recent experimental findings where we introduce some theory, and that theory explains the experiment. So with that, I'll move to the plan of talk. So first, I'll discuss concept of different times that can be measured experimentally and theoretically in case of protein folding unfolding dynamics. Then I'll briefly describe the experimental observation on these transition paths, and later on I'll survey brief of literature on this theoretical interpretation available for this experimental observation, and then we'll go for our theoretical approach, and again I will come back to the experimental evidences that actually suggest that our theory is valid, and finally I'll end up with a new approach that we introduced in this transition path dynamics. And so with that, I'll start the concept of different times. And so like there is a schematic picture of this barrier crossing dynamics for the protein folding and unfolding, and where you can see the first concept of time that I mentioned, it is a waiting time. So experimentally one can measure the waiting time, and theoretically also one can measure the waiting time. So what is waiting time? Waiting time is that when biomolecules make a transition, before that it wait for some time in any of the minima or folded or unfolded minima, and that time you measure and is known as the waiting time. So for many trajectories you measure many times and you plot a distribution, and that will be waiting time distribution. Then another thing comes, like for waiting time, one important thing is that like the transition is considered as a instantaneous transitions. Okay, so the main time is involved for this wait, and then transition is instantaneous. But then again comes with the first passage time. So first passage time is like is a you start from a point of B where a trajectory is shown there, and one trajectory starts from A and it's coming back and forth to the initial position, then it makes to transition to another position which is B. In barrier, it is shown as a first passage time with a red arrow, while like you start from a minima, you go back and forth, and you ultimately make a transition and you reaches to a target. And you measure the whole time, that is a first passage time. And however, this first passage time and difference between the first passage and waiting time is that like when there is multiple intermediate states, you just wait in the intermediates for many times. So there is no specific waiting time for any of the well, and you have the all the wells will contribute the waiting times case, and it will ultimately give us the first passage time. So both from the first passage and waiting time, one can calculate the folding rates and times, and like which is like very commonly known. However, we try to explain here the transition part time, which is like 1,000 times, almost in time scale is shorter than waiting and first passage time. So what is transition path time? You see that in the barrier region there is two dashed line, and in the barrier region two dashed line which is shorter than the time spent with the minima, because it is very short region around the barrier or the barrier top where the molecule makes a transition from the transition stage. So in this case, in the trajectory, it is shown as a first passage time for the full region where like there is molecules make back and forth plus a transition with to the top from A to point B. However, for the transition path, there is like dashed line showing the trajectory is directly transit from A to point B. So there is no coming back on the point A, or the molecule will not come back after reaching from the point B. So both the cases there is the absorbing boundary on A and on the point B. So that's why transition time is very short and it is very difficult to measure experimentally. And one need very finer time resolution to do that. Recent experiments enable the measurement of the transition path, where like Neupan et al., where it is a Woodside group experiment, where picture A it is showing that the barrier crossing dynamics, a trajectory is moving from one minima to other, and in the picture B it is showing the experimental setup, where like two blue beads are attached on a, and the laser trap is focused on the bead, and then with a polymer tether, a DNA or protein handle is attached with the tether. And now this laser creates a tension, and that's why the molecule stretches, and one can get extension versus time plot. So the molecule can go from the unfolded state to folded state because of that tension. So the measurement is done on the equilibrium where that force is such that the population on the unfolded and folded state is almost equal. And then the measurement shows that extension versus time trajectory on the picture C, where you can see that most of the time on the equilibrium trajectory that U is unfolded state and F is folded state, the trajectory is going back and forth and oscillate between unfolded and folded state. However, there is a short region of the time where like they move from the unfolded state or the folded state. So it is a very short time that I mentioned earlier also. So then there is a zoom in picture of this trajectory from C to D, where it is shown that initial cases the trajectory is going back and forth to the initial position, then it moved to the another state, and in between there is x1 to x2 two region is chosen such that that trajectory will not come back from one point, the initial point, and to the final point. So it will go from x1 and it will ultimately reaches to x2, and there will be no coming back from x2 to x1, or if trajectory goes from x1 to the bottom side, then it will not go to upwards. Okay. So with that in mind, the experimental setup and the trajectory, one can measure the time required from going x1 to x2. And for different trajectories, you can measure several times this x1 to x2 time, how much time it requires. And you plot a distribution or histogram of these trajectories or for the time, and then you can get the probability distribution for this transition path time. So this path is known as the transition path, and the time requires to go from x1 to x2 is known as the transition path time. So it's showing that the experiment histogram showing with a solid line fit, which is the analytical result, that is the expression for this, and it's showing very good fit of this transition path time. And where this long time is zoom in on the top, where it is showing the long time tail, which is exponential, and it is also well fit with the available theoretical result. However, the problem with the theoretical result is that only open boundary condition result is available analytically. So the expression is available, you compare with the experimental transition path time distribution, which is absorbing boundary, but you are using a open boundary condition expression. So what is the problem with open boundary condition? Open boundary condition is that the trajectory can go back and forward. That's what it's giving the long time tail. So if we consider absorbing boundary, trajectory will be shorter, okay, and the time requires will be smaller. So that's why there will be a discrepancy between theory and experiment in this case, although it is showing very good fit. So there is another discrepancy that I first mentioned. That first discrepancy is that absorbing boundary condition. If you consider, then with a smooth parabolic barrier, that is minus half kx square. If you consider that it is go over that minus half kx square barrier, that is a parabolic barrier, then the long time tail will not be explainable for this case, because it is considered as an open boundary condition theoretical result. So the second discrepancy is that with the fitting parameter obtained from the analytical theory and with the experiment, experimentally it was earlier obtained that for these two experiments they did with a DNA hairpin and PRP protein, for one case DNA hairpin they obtained the barrier height as a, the full for unfolded to folded barrier height as a, for hairpin it is about 9 kbt, between 9 to 10, and for PRP it is 4.5 kbt. Kbt means it's a thermal energy. Okay? So what happens, like when one fits with the available analytical results, then one get that for hairpin the barrier height is about 1 kbt only, and for hairpin it is 0.5 kbt, which is far less than the experimental estimate. So which is wrong. So what could be the possible reason for this kind of discrepancy that we get from this experiment and compare with the analytical theory? So there are mainly two problems. To explain, there could be many possible reasons. So basically in theory, the available theory, most of the theoretical expression people use as a overdamped limit where friction is large. So this, in this case it is shown as Langevin equation, while like the first term is the inertial term and the second term indicates that derivative of potential, which is force, and the on the second term, that means the on the right hand side the first term, and the right hand side second term indicates the friction coefficient, and the last term is the random force. Okay, so this Langevin equation, when one considered the overdamped limit or large friction limit, then the left hand side term with left hand term, that is m d2x dt2 will be dropped. So what could be the consequences? The consequences is that it hardly affects the dynamics, because most cases on the experimental conditions or simulations, computer simulation, you can see that it affects on the very short time scale. Even the time step, it is shorter than the molecular dynamics time scale. And another thing is that if you include the inertia term, that system propagation will be faster, okay, because the viscosity or friction will be less, so the system will propagate faster. So we need a long time tail. That means this is giving the opposite scenario. That means if you consider on inertia term, the system will give the shorter distribution because system propagates faster. So this is not the possible explanation. So the second explanation could be like memory effect and inertia effect combined. So in the same generalized Langevin equation, if you use the memory kernel, which is k t minus t prime, and if the memory acts in this dynamics, it could be a could give a longer time tail. People thinks that, but in real scenario, when like most theoretical formulation use as a power law dynamics for this memory kernel, where kt is proportional to t to the power minus alpha, or where alpha is greater than 0 and less than 1. When alpha is 1, it is completely Markovian and go back to the original Langevin equation that I showed earlier, and it is for the power law dynamics is between 0 to 1. So what happens if we consider this power law dynamics, which is mostly common in theoretical and experimental conditions people use? The stronger correlation in the random force actually facilitates the motion. So if the motion is facilitated, again the propagation will be faster. Then we'll not get the longer time tail. We'll get the shorter time scale than the experimental time scale obtained. So another problem is there that we need to get this memory kernel experimentally. So or from simulation. So however, there is one technique is popularized by Bruce Barnes long time ago, 1990s, where they used to estimate this force from the force autocorrelation function, these frictional kernels, memory kernels. However, the time scale for this is far from being the transition part time scale, which is microsecond to millisecond, and that's why the estimation of this memory kernel will not affect the dynamics. Okay, so the next reason could be the dimensionality, because many people thinks that protein folding or unfolding is a multi-dimensional problem. So so if there is another dimension available, molecule can stay to another dimension for some time, and that's why you are getting a longer time till. That could be another explanation. However, like Makarov and co-workers in 2020, they introduced one parameter that is known as the correlation coefficient C, which is defined as the standard deviation S by mean time. That is a T average. So when the standard deviation and mean time is greater than 1, then it should be one should use the multidimensional aspect of the dynamics, because from any distribution or one dimensional distribution, one dimensional potential, one cannot get the correlation coefficient greater than 1. So for one dimensional any kind of potential barrier, the correlation coefficient will be always less than 1, but one can get correlation coefficient is equal to 1. So if the distribution will be broader, the correlation coefficient will be larger. Okay, so as the standard deviation will be larger. For 1D well, the C value one can get is equal to 1, exactly equal to 1. Why? Because for 1D well, instead of the barrier crossing dynamics, it will be escaping dynamics or waiting time dynamics that I mentioned earlier. So it will be a waiting from the well to outside. Okay. So the waiting time distribution, we know for a single well, if it is a Markov process where no memory effect is included, then it will be like a exponential. And for this exponential waiting time, it is pt is given as k into exponential e to the power minus kt, where like k is the rate for forward and backward processes, sum of the rate for the forward and backward processes. So from this, using this distribution, if one calculates the correlation coefficient C, one can get easily equal to 1, because it's from this distribution one can calculate easily to the first and second moment, and one can get the value equal to 1. So another important thing could be that effect of the bead, that probe we are using, people are using that during the transition path experiment. So we are seeing that the dynamics of the protein folding and unfolding. However, the beads is all we are measuring the movement of the bead. So bead can affect the dynamics. Okay. So however, the mean time, if you consider only bead and the tether, then the mean time will be 5 microseconds. However, the transition path mean time for one DNA hairpin is about 30 microsecond, and for another PRP protein will be millisecond. So beat can hardly affect the dynamics. Beat affects the dynamics, but not that much that will create the long time tail. And another important thing is that to include the effect of the bead, we need to introduce another coordinate on the dynamics, which will be the coordinate of the bead. So it will be a two-dimensional problem. One will be the coordinate of the protein folding and unfolding, another will be the coordinate of the bead. So then we'll come back to the second, again the same problem with the correlation coefficient. We first check whether there is a correlation coefficient is greater than 1 or less than 1. If it is greater than 1, then multi-dimension problem is required. If it is less than 1, it is not at all required. Okay. So the only solution for this problem is the barrier shape. So the barrier shape can affect the dynamics. So to investigate this, we consider the overdam limit of the Langevin equation. We drop the first term of the Langevin equation, which was m d2x to dt2, and we end up with a diffusive dynamics with the presence of du dx, but u is the potential, which is the another version of the Spolozowski equation. Now, with this delta correlated noise, that is 2 zeta kbt and delta correlated function for this random force, one can calculate the time requires from going from one point to another point. So we calculate with the same way the experimental people did, that we start from a boundary minus a and we go to a, and where the intermediate state is around 0, and then we calculate how much time requires from minus a to a, and we plot a histogram, and then we take a normalization. The denominator actually indicates the normalization of this histogram, and we get the transition normalized transition path time. And similarly, one can calculate the moments from this first moments or second moments from this distribution in order to compare the mean time with the experiments. Okay, so we first start with the anharmonicity and asymmetry effect of the barrier. So we consider different type of barrier where like two linear term of the potential is known as the cusp one, blue one, then parabolic which is minus half kx square, quartic which is minus kx to the power 4, and hex x to the power 6 type of potential. And we see that from going from parabolic to anharmonic potential, we hardly see any difference between that normalized potential that we early explained, that this one-dimensional case while like smooth parabolic potential cannot explain the long time tail. And you see here that in this case the long time tail in this experimental, the symbols are represent the experiments, and in this case long time tail is not explainable from this theoretical uh, transition part-time plots, even if you consider the anharmonic effects on the barrier. So then we move with a asymmetric potential, where like potential is consist of a parabolic uh, for the some part and with a linear type of potential at the end. Because in experimental cases you always not get the smooth potential where this potential is a harmonic one. You can get a anharmonic potential with the asymmetry on the position of the barrier top. So with this asymmetry effect, we see that even if you consider extreme asymmetric limit where this, you see the red potential which is extreme asymmetric compared to the blue one, and one see that one can get the dynamics is hardly affected, and there is a long time tail improved but not that much to compare with the experiment again. And we also mentioned about the correlation coefficient earlier, where correlation coefficient, if it is greater than 1, it is a multi-dimension problem. If it is less than 1, it is a one-dimension problem. So in both the experiment, we compare here with only one protein, only one molecule, which is the DNA hairpin, and we didn't show here PRP protein. We'll show later PRP protein. Both the cases, the correlation coefficient is far less than 1. So there is no question of multi-dimensionality. So next, we'll try to explain by reducing the diffusion coefficient, because if the diffusion coefficient is slow, then dynamics can be slow. Then first thing we'll try with a reducing the barrier height, where you can see from the blue to red, by reducing the barrier height by keeping the diffusion coefficient same, one can see that there is a little bit improvement of the long time tail of this transition path. However, this is not physical, because as I mentioned earlier that lowering the barrier is not a solution, because the experimental estimate already is higher than the theoretical estimate earlier, which is 1 kbt, and the experimental for one protein it is 10 kbt, another one is 5 kbt. So the reduction of the barrier height is not a solution, because why reduction of barrier height increases the long time, there is a reason. We think that otherwise that when the barrier is more steeper or the barrier height is larger, it requires much more time to cross the barrier. But in transition path kits, it happens the opposite. If the barrier height is more or larger, it requires less time. Why? It is a surprising thing, because when we consider for transition path only successful trajectories that goes from one point to another point. Okay, so if one considers the successful trajectories, then the main problem comes with the energy conservation. If the barrier height is like uh, getting increased, then what will happen, that it requires much more energy to cross the barrier. Okay, so much more kinetic energy to cross the barrier. So kinetic energy will be higher and the velocity will be obviously higher, and that's why it requires shorter time. So then we tried with lowering the diffusion coefficient, and diffusion coefficient is rescaling the time towards the right hand side, and that's why giving the long time tail. However, the maxima of the distribution will be also shifted to the right hand side, and that is giving the wrong distribution with respect to the experiment. So the diffusion coefficient reduction will not be also a solution. Then we try to distinguish with theoretical point of view. One can determine what the barrier shape from the mean time data, whether it is possible or not. So we tried with different smooth barrier like a cast, parabolic, and quartic and hexic, and all the cases we plot mean time versus width of the transition path, and we plotted mean and standard deviation. On mean is represented as a circle, and the standard deviation is represented as a square. And for all the barrier cases, we varied the width, where below it is shown as the width of the transition path as a 2a from minus a to a. So it will be 2a. If a value is 1, we cover the full transition path. If a value is less than 1, then we will not cover the full transition path. So with changing the extent of the barrier, we wanted to see whether one can distinguish the properties of the barrier from the mean time. But for even for the full transition path case, we see there is hardly any difference between all the barriers for the mean time, and there is no distinction between these mean times. Why? Because the one cannot distinguish this, and experimentally it is also not possible, because the experimental error bar is 10% for the DNA hairpin for the mean time, and for the PRP protein is 20%. So experimental error bar is higher than the difference between the mean times available for this differences between mean times for this all the barriers that we use. So then we see, as I mentioned, that mean time with the increases with the increase in width. In this case, you see that width is the increasing width, mean time increases, which shows the reverse trend with the barrier height that I mentioned earlier. So then we come up with a solution that if there is a intermediate states or trap in between or well in between a barrier that I mentioned earlier, well can give the longer time distribution, because the particle or the biomolecules can stuck sometime for the intermediate state and giving rise, the trajectory is showing here, and giving rise to a longer time for this reaching to the target. So we use two type of potential. One is the cast barrier well, which is contains of straight lines, and another is piecewise quadratic, which is made of three regions of harmonic potentials. And with using this, we get another parameter, which is well depth of this type of potential. Initially, there was a barrier height. Now there is another parameter, which is well depth. Now, varying the well depth, if one plot the mean time for these two type of potential, one can see there is a clear distinction between the cast barrier well and piecewise potential at longer or larger well depth, and both shows a quadratic behavior, which was not available from the analytical calculation earlier. So, for large well depth, one can distinguish between different potential shapes. So, as I mentioned, that trapping of the particle in the well can produce longer time tails. So, we see from this both cases, from the cusp one and the piecewise quadratic one, reduction of the the increase of the barrier or increase of the well depth from 1.3 to 2.3, and here 1.9 to 2.9 actually gives the exact fit with the experimental result, and thereby reproducing the longer time tail. So uh, we also get, importantly, here the barrier height about 3 kbt. We fix the barrier height about 3 kbt for DNA hairpin, and another protein we also fit the exact fitting we got for the both potential about barrier height is 2.5 kbt. So the presence of trap not only explain the longer time tail, which is showing the exact fit with the experiment, also the barrier height is improved about 3 kbt. And from the earlier experimental estimate, we show that for DNA hairpin, that full for folded to unfolded minima is the barrier height is 10 kbt. However, the transition path covers, as I mentioned from the first slide, the transition path covers the only the barriers, the nearest region of the barriers, which is almost half distance between the folded and unfolded minima, and that's why the 3 kbt estimates is more realistic than previous estimate of 1 kbt. And for PRP protein, we get about 2.5 kbt, which is more realistic than previous estimate of 0.5 kbt, where the full potential for full potential case the barrier height is about 5 kbt. Next, just after we published our paper, the same experimental group came up with a final resolution experiments, and then immediate after our paper published, they showed that there is an intermediate region. There would be many pauses on the transition path where trajectory stays for some time, and the pauses are shown by these bars of the pink colors bars and or the purple color bars. And then one can see between the x1 x2, which is the transition region, uh, there is some trajectory that are showing these pauses, and out of two trajectory they concluded out of two trajectory, one trajectory showing pauses and almost 50% trajectory showing pauses. And that's why it is really important, and they estimated the pauses, the well depth of these pauses. They uh, explained that there could be multiple minima in between, although we started with a single minima inside the barrier cases, or single well, which is simplistic version of this model, but they obtained that there could be a multiple uh, well inside the barrier, which could be of well depth of about 0.9 to 1.4 kbt. Then they plotted the pause time distribution, which is known as the dwell time distribution or the waiting time distribution that I explained earlier. And they try to fit with single exponential, which is that straight line which is pale blue, and then double exponential, which is deep blue, and red one, which is the rugged energy theory of the Walliness, where they consider that potential energy is rugged, and there could be a depth of multiple depth are there on the potential, and that's why giving rise to a distribution which is logarithmic, and one can get a good distribution. And they also conclude at least there are two types of wells are there, micro wells are there inside the barrier, which is giving rise to that by exponential behavior. If there is only one well, it should only give the single exponential that I mentioned earlier. However, the simplistic view is that at least two types of micro wells are there. So keeping in mind that, and we then try to explain that diffusive dynamics theory using the microscopic point of view. So what is the microscopic point of view? In diffusive dynamics, one uses the Smolosowski equation, which is a one-time limit of high friction limit of the Langevin equation. Then one can start with a master equation model where that barrier region is discretized by many microstates. So one can question how many microstates are needed, or how many micro wells are needed to justify these uses of the Smolosowski equation, because in some cases where the rugged energy landscape are there, the Smolosowski equation will not be valid because the potential is not continuous. So what could be the solution? Then only solution will be the master equation approach or microstate approach that one can use. And the question will be how many microstates are needed to justify the Smolosowski equation or the experiments. So the simplistic model Van Kampen used in his non-equilibrium book, he used a nearest neighbor jump model between the microstates, and he explained for any arbitrary potential one can ultimately reach to the Smolosowski equation. And to in order to consider that, we come up with a compare with the diffusion of the Smolosowski equation with the master equation model. But diffusion equation is that this is the discretized version of this Langevin equation, or this is known as the Brownian dynamics or stochastic differential equation, where this random force term is a stochastic one, and one can get the solution of this discrete version of the equation, and one can compare with the master equation model. So how this master equation model is considered? One can discretize the barrier region with different microstates, where the different color indicates the different realization of this microstates, different set of microstates, and one can get the forward jump probability using this equation and backward jump probability using this equation, where J plus up indicates the energy of this forward barrier, and J minus half indicates the energy of the backward barrier, and J indicates the energy of the microstate. So one can get from the forward and backward jump probability from this equation. And the rate of the forward and backward jump can be given by this Arrhenius type equation, and where this is Arrhenius type coefficient, which is connected with the friction, and there is again this energy of the microstates or microwaves, and this is the barrier's energy, J plus half and J minus half. So if one uses this type of microstate models and start from this position, the extreme left position of this microstate, and wants to go to the extreme right position of the microstate, then this equation will be used. Using the depending upon the probability, you can choose the forward or backward jump. If you start from the extreme left, you go another left, then the trajectory is cancelled or not considered. If you are going forward and ultimately reaches to the extreme right, then trajectory is considered. And the time is the duration of the between the times is between the jumps is considered as logarithmic of this. This is a random number which is between 0 to 1, which describes the probability for this jump, and this is the forward to sum of the forward and backward depth. Why this equation is used? Because I mentioned for a escaping or waiting time from this any well is considered is exponential. The time distribution is considered as exponential for the Markov process, as it is considered master equation is always this Markov. So that's why the distribution, the time delay or the distribution of the time delay is exponential. So the time is propagated as t plus delta t, and when the particle reaches the final state, the time propagation stops. So this is known as also the kinetic Monte Carlo simulation. Different people call it differently. So biology people also consider this type of simulation as a Gillespie algorithm. So using this, sorry, uh, using this, we consider, as I mentioned, for different realization with a small microstate models. With a 5 microstate I started, and 5 to 11 to 21, 31 microstate we used. We tried to go to the continuum limit where many microstates are possible, and where the the barrier is barrier is constitutes like that the top of the barrier touches the parabolic barrier, where the energy is same equivalent to the parabolic one, where the microwave's energy is random component and chosen from this Gaussian uh, random component. And if one use this smallest microscopic model of 5 state, then one compare. Here is 3 realization I shows what 3 different sets of microstate. It's showing with the diffusion or Smolotowski equation is showing it's not good fit but very close. However, one goes to 5 to, I'm not showing that others microstate as 11, 21, I'm directly showing the 31 microstate case, where for 3 different realization 1 to 3, one get exact fit with the diffusion equation. So because we are going towards the continuum limit, so that's why we are needed at least dozen of microstate to explain the Smolotowski equation or to justify the Smolotowski equation, and this is the master equation for the forward and backward jump rate. And to then we compared the mean time for the diffusion equation and the master equation. From this on the reduced unit for diffusion equation we get 0.42, and from master equation for different microstate we tried to get as close as possible. Out of 100 of realization we tried, and we see we can get closest from this get close from this diffusion equation for different cases, like even for 5 microstate, 5 number of microstate we get close to this diffusion equation. And however, one if one compares with the highest value of this or good fit with the mean time one takes and compare the shape of the distribution, then the two option comes with, one is the Pearson correlation coefficient, another is mean square error. If one the Pearson correlation coefficient is 1, then it is fully correlated, and if it is 0, it is not correlated. Two data sets, two plots. For the mean square error, if it is two data sets are same, then the mean square error will be 0. We see that from 5 to 11 to 21, 31, that Pearson correlation coefficient is going towards 1 and saturated for 21 to 31. So that's means 21 my number of microstate is sufficient to explain the diffusive dynamics or the continuum behavior. And for the mean square error also is showing the similar behavior and is going towards 0 for 21 microstates. Then we compare with that the similar model with a barrier well inside a barrier with a piecewise quadratic behavior, and we compare with the diffusion equation or Smolensky equation with this master equation models, and for different realization for this DNA hairpin and PRP protein with uh, 3 kbt barrier height, and for DNA hairpin and 2.5 kbt barrier height for this PRP protein. And using this, one can get exactly fit and even the better fit for this DNA hairpin and PRP protein from this 20 at 1 microstate model tied with 11. 11 is the is also giving good fitting, and so from this one can conclude that master equation shows good agreement between the experiment and the Smolensky equation, which is designated as AC, and also master equation models validates the claim of the presence of the trap in the transition path, and also it confirms the barrier height about 3 kbt, which is more realistic than previous estimate of 1 kbt. Then we compare with the mean time with the theory and experiment. For this experiment, the DNA hairpin, as I mentioned, which is reduced unit is 1.62 and error bar is plus minus 0.12, and for PRP protein is 1.5 and zero point 0.3, and then diffusion equation gives very close to the experiment for both cases, and from the master equation using 11 and 21, we see both cases we can get close as much close as possible for this experiment by using the same barrier height and well depth that we used earlier and discretize the barriers. However, the important thing is that from 11, if you use 11, you need to try for many realization. If you use 21 microstate, almost all the realization, all attempts will give you the very good numbers. As I mentioned, you are going towards the cardinal limit, so that's why. So uh, with that in mind, I want to go to the last part of this talk, and the last part will be like a conceptual part, where like we think that as I mentioned, that we think that if the barrier height uh, is larger, it requires larger time, but in transition path case, it is reverse, and the same another misconception about this barrier crossing is that. If one consider the barrier, people think that the climbing of barrier requires much more time than going down the barriers. So we try to clarify this misconception, and to clarify this, we start with a simple available theoretical explanation, while like it's another time which is used as a translocation time, which is used for the passage of a particle through a channel or a tube. Okay, so you start from the left, you are going right, or you can start from the right, you can go to the left. Where the similar absorbing boundary at both ends are used, similar to the transition path cases, and you measure the time request for each time you go from one side to another. So the translocation time is defined as the flux by the normalized flux with respect to time, and the flux is like how many particles that are arriving to uh, the targeted boundary. Okay, so this group by the Berezovsky and et al. They observed that whatever the profile inside of this tube, or if there is no potential at all inside, one can get the equal time for going towards if you start from left to right or start from right to left. So whatever the profile inside, you can get equivalent times for the both passes, both cases. Okay, so although the translocation probability at the both end will be different, because depending upon the profile at the both end, or the the area of the both end, the number of trajectory will come out will be different. Okay, so the translocation probability will be different, but the time requires, or the time distribution for one side to another will be equal. Why this is equal? This is actually because of the detail balance condition, uh, or the time reversal symmetry that is that they use to prove this, the from passage of one particle from one side to another. So what happens with the uphill and downhill for the barrier crossing? So we tried with the same with the diffusion or Smolensky equation and the master equation model. for the uphill and downhill with same type of model. And with uphill and downhill, in this case, is already shown that uh, that for the diffusion equation, both Smolotowski equation cases uphill and downhill is giving the similar type of distribution, and the mean time is also same of the both cases. And for the master equation cases, there are many realization are tried with the smallest possible microstate. With 5 microstate we tried, and then we see that these different colors, blue and reds, are used, where the same line style indicates the up and down time for the one set. Different line set is different set. So for both the cases, for the 3 different cases, we see that up and down times are same, the distribution are same, and giving the similar mean time for the up and downs. So why this is happening? Because of the same, the detail balance condition is maintained, and the time reversible symmetry is maintained. Uh, Although the up down probability, as I mentioned, the translocation probability is different earlier cases. In this case also, the up down probability will be different. The number of trajectories coming up will be less than that of the number of trajectories coming down. This situation can be different only if the system is non-equilibrium. That kind of a system, if it is a molecular motor kind of a system where the forward propagation is only allowed, and the non-equilibrium system can be constructed using an additional noise term on the Langevin dynamics. Then one will break the detail balance or time reversible symmetry, and one will get different time for the forward and backward process. So with this, I conclude that presence of trap or well inside the barrier is the only possible explanation for this long time tail in the transition path time distribution, and the transition path barrier height of 3 kbt, which is more realistic than previous estimate, where one get 1 kbt or zero point 0.5 kbt for 2 different cases, DNA hairpin and PRP protein, and at least dozen of microstate is required to justify the use of the Smolotowski equation. And then another important thing is that master equation is more advantageous than that of the Smolotowski or diffusion equation, because in some cases you can get the potential which is rugged, which is not continuous. Then you cannot use this approach of this Smolotowski equation. And finally, we proved that even for the barrier crossing, the up down time is equivalent, and this only will break if the detail balance condition will be broken. Then for example, for the non-equilibrium cases, where like a molecular motor cases. And with that, I thank you everyone for attending this talk.
Thank you, Rajesh, for your wonderful presentation. I think students might have some questions for Rajesh. Any questions? Yes.
I want to, what is it mean to have different realizations of some?
Okay, so just try to go to the slide, then it will be more clear. So different realization means, like if you see this 5 microstate model, where like blue, green and red, 3 different are used, where you see the top touches with the barrier, parabolic, smooth parabolic barrier, which is minus half kx square, where however the depth of the wells are different, or this is random. Okay, so random depth of the wells are chosen using a Gaussian random number generator. Okay, so for each different times you generate different random number set, and you will get different depths of the micro depending upon how much we use the mean of the well or the variance. Okay. So you each time you will generate different random numbers, and you will get different type of micro wells.
And that means each realization means different energy of the configurator of the.
Yes, yes.
So, any more questions? Any questions from the online?
Yeah.
I think,
Yeah, can you hear me?
Yeah.
Yes, come on.
Hi, Rajesh. Yeah, very nice work, and very systematic, thorough investigation. So, I had couple of kind of clarifications to ask. So, very nicely have compared the different possible reasons for this long time distributions. You have eliminated all these aspects. So,
Yeah.
I just wanted to clarify that if we just look into the effect of ruggedness itself, just a bimode, I mean, two minima system like your parabolic barrier with ruggedness. So there will be multiple minimum in this system.
Yeah.
And you said that this is not enough for reproducing the long time distribution, long
Uh,
tail.
No, no, I didn't tell that ruggedness will not be enough because we used a very simplistic version of the ruggedness. That is, we used only one well inside the barrier. And next, I showed that immediate after our paper get published, then experimentalists come with a ruggedness model, while like they showed there will be multiple minima that could be possible for the long time tail, which is about 1 to 1.5 kVt. So our approach was more simplistic version of this ruggedness. So if one wants to use the ruggedness model, one can also get the long time tail, but we don't know how much long time tail you will get from this. And
Right.
but you
So
can
what
use
I
the
was
master equation model to explain the ruggedness.
Yeah. What I was trying to get at is, so the ruggedness has an energy scale associated with these small undulations.
Yeah.
Now, is there any kind of a critical depth required so that, so what I'm trying to get at is there can be just small noise in the barrier height that can also, you can call ruggedness, little bit of undulation, and there should be another deep minima, that is your intermediate state. So
Yeah.
I'm just trying to distinguish between these two kinds of models with small noise with small minima versus having one deep minimum, which is your intermediate state. Do
Yeah.
you
Like
see some qualitative difference between these two scenarios?
yeah, there are like I talked with different experimentalists.
Hmm.
Like there are two possible scenarios. One experimental group from it is the University of Switzerland. His name is I forgot what is his name. So he Benjamin Schuller. He told that they found an intermediate. Okay,
Hmm.
so the intermediate is well depth that you can found the intermediate uh, for this type of experiments. And another by the Woodside group himself, they claim that instead of one intermediate, there would be a ruggedness, and you can not call it as a noise because it's not about zero point 0.5 kBt or something less than that. It is more than 1 kBt is even possible. So in that sense, it is ruggedness, but maybe if experimental resolution is improved far, then you can recover the intermediate also. So we don't know the exact case what is happening.
Right. And another thing I would like to just point out or just discuss
Yeah.
that already mentioned about this issue of dimensionality, that
Yeah.
having higher dimensionality introduces many other pathways. So that in that context, in
Yeah.
your 1D model,
Yeah.
uh, all trajectories must go through the intermediate. You cannot escape the intermediate. But in a higher dimensional space, there can be multiple pathways. Let's say in some trajectories can even directly go, some
Yeah.
can get trapped in intermediates in a
Yeah.
higher dimensional space. So there can be a like mixture of pathways.
Yeah.
So in that case, there will be this. So 1D and multidimensional things can have a very different pictures qualitatively.
That's true.
Yeah.
So to determine that, I told that Makarov group had introduced a correlation coefficient. So first you need to go to the experimental distribution and you need to check whether the correlation coefficient is greater than 1. Then multi-dimensional model is required, and if it is less than 1, one-dimensional model is sufficient to explain the experiment.
Mhm. Okay. Yeah. Okay. Thank you. Thank you. Very nice work.
Thank you. So, any more questions? Okay. If there is no more questions, so please join me in thanking the speaker.
Thank you very much.